Hello and welcome to week 9. Time flies when you're having fun. So this week we're going to talk about translation and proteins, and we're going to be focusing on chapter 14 from our book. So just to continue from last week and just to refresh your memory, translation is where we're going to be taking the mRNA sequence and we're going to be turning it into something useful for the cell. In this case, it's going to be proteins, or a series of amino acids. So translation is going to be where we're taking many different amino acids and we're going to polymerize them or put them together into polypeptide chains. Remember, this is just a long chain of amino acids. So in order to do this process, you need several different components. First of all, you're going to need amino acids. Amino acids are absolutely required. They're going to be a big part of this. You're going to need messenger RNA. Remember, this is what is made at the end of transcription. We will have mature messenger RNA. It will leave the nucleus in eukaryotic cells, go to the cytoplasm where it can then be translated. The location of the translation process is at ribosomes. We'll talk more about them in a minute. And the last component that is required is transfer RNA, or tRNA. So let's focus in on ribosomes first. Ribosomes consists of proteins, the ribosomal proteins, as well as ribosomal RNAs. And we're going to call these rRNAs. Ribosomes have a large subunit and a small subunit, and they're going to assemble around messenger RNAs. These rRNAs are going to perform important catalytic functions associated with translation, so they're absolutely required for this process. One of the things that they're going to do, or the ways that they're going to do this, is promote binding of the different molecules involved in translation and just to really help fine-tune this whole process. So if you're to look at the ribosome, they're very similar in both prokaryotic cells as well as eukaryotic cells. The only real difference is size. So in prokaryotic cells, you're going to have a large subunit, okay, and you're also going to have a small subunit. And what you'll see is that this is going to come over to here. The mRNA is going to be read like a ticker tape through this assembly. Eukaryotic cells, very similar structure, just slightly larger in size. We actually do know all of the components of these. We know the proteins that are involved, we know the RNA nucleotides that are involved, and so on. So let's talk a little bit more about the tRNAs. tRNAs, some people call them adapter molecules, and the reason for this is that they're going to literally adapt or help to translate this genetic information that is present as specific triplet codons in the mRNA. And the way that they're going to do this is they have what we're going to call anticodons. Remember, mRNA is going to have codons. We use the codon chart. We can look up to see which particular amino acid that would eventually be coding for. The tRNAs have anticodons. It's going to be complementary to the codons. And so it's going to complement the mRNAs. And they're going to temporarily be binding to one another. And this is the reason why it has to be complementary to one another. The other major important function that they're going to do is that they're going to bring in the corresponding amino acid. Please note, again, in the codon chart, you're looking at the mRNA sequence. The tRNA is just going to be complementary to that sequence. We don't use the tRNA sequence to figure out what amino acid that tRNA is carrying. tRNAs in general are pretty small, they're pretty stable, about 75 to 90 nucleotides. They're actually transcribed from DNA, as is anything else. They're going to contain post-transcriptionally modified bases. And some of the things these are going to do is to help to enhance hydrogen bonding efficiency during translation. You may be familiar with the structure of it, but the 2D structure, if you were to look at it, looks somewhat like a clover leaf. Very, very appropriate as we're coming up on St. Patrick's Day. As you can see here, there's somewhat of a clover leaf. On one end of the tRNA, this again is the 2D structure, you're going to have the amino acid binding site. You can see it is single-stranded, although there's some double-stranded bonding through various parts here. The other end of it, the opposite end, is going to contain the anticodon. This is what is going to be complementary to the mRNA. There are several other loops to the side, what we're going to call a variable loop, and then the 5' end of the tRNA. There is a hydroxyl group on the 3' end as always. There's a phosphate group on the 5' end as always. And there's a sequence here called CCA. This will become more important as we talk about it in a little bit. If you were to look at the structure of this in a 3D conformation, it doesn't look very much like the 2D conformation. But the same overall sequence is there. You can just use your imagination to imagine some of these coming forward or back away from you to form this 3D structure. Okay. So like I said before, it's going to have these tRNAs are going to have an anticodon, going to complementary base pair with the mRNA, and that the end of that CCA sequence is on the end of all the 3' end of these tRNAs. Okay. One of the important components is that they're going to be carrying amino acids, the respective amino acids corresponding to whatever codon that they would be attracted to. The way that they do this is that they have to be activated first. You have to charge them. There's specific proteins or enzymes that are involved with this process. We'll call them aminoacyl tRNA synthetase. As you might imagine, there's 20 different ones because we've got 20 different essential amino acids. Okay? They're very specific. They're only going to recognize their amino acid. So the basic process of what's going to happen, you've got your aminoacyl tRNA synthetase, you've got an amino acid, you've got some ATP, you're going to combine them together and you're going to charge it. You're going to activate this enzyme complex. It's going to be loaded up. You've got an empty tRNA here. You're going to, the enzyme will basically add that activated amino acid to the end, and now you have your charged tRNA. This little X here is just to designate that the amino acid could be any of the 20 amino acids. And that little AA is just for amino acid.